Hello everyone, welcome to my crash course on vaccine design. So I'm going to start off with a basic definition of a vaccine. In simpler terms, we can define a vaccine as a substance, either a dead microorganism or a live attenuated organism that can trigger the body's immune system without exposing the body to the full strength of the disease. So of course, a dead organism is not going to have any impact on the body. A live attenuated organism, that is just a fancy way of saying a milder version of the organism. It also is not going to have as much impact on the body. Um, what is the purpose? What is the purpose of all of this? Basically, vaccines are a way of preparing the immune system for a disease so that if we ever encounter that disease in our daily lives, we would not get sick. I am going to briefly mention the 6 phases of vaccine development, and this breakdown is provided by the Center of Disease Control, CDC, and they've basically narrowed it down for us. So as you can see, the 6 phases are exploratory, preclinical, clinical development, regulatory review and approval, manufacturing, and quality control. So this is a lot to talk about for this video, so we are just going to be focusing on the preclinical phase. If you want more information on any of the other phases of vaccine design, feel free to check out the Center of Disease Control website. There's a ton of information available. Before we proceed, I would like to do a little bit of a vocab checkpoint, and I like to do this using a brief analogy. So as you can see here, we have a bad guy, a bad rat, and he's always wearing the color orange. He is the pathogen. The pathogen can be a bacteria, a virus, or a microorganism that can cause a disease. So an antigen would be a piece of his clothing. So either his hat or his orange shirt, that would be the antigen. It is unique to this bad rat, and he always has it on. The police officer would represent the body's immune system, and the police officer's baton would represent the antibody. So, if the police officer were to detect the bad rat because of his orange clothing and hit the rat, poor guy, taking him down, it would be equivalent to the same way our body's immune system acts to protect us against pathogens. Finally, the main purpose of this video, the preclinical phase. As you can see, it has 5 steps within it. Most of it occurs in a lab setting, and it is usually private labs. So the first step in a lab is to generate the antigen. Scientists isolate the antigen from the pathogen, so basically like removing the orange shirt from the rat, and then they make tons of it. They make tons of it using highly replicative cells. The second step is to release and isolate the antigen. Scientists must find a way to separate the antigen from the cells that they were grown in. The third step is the purification step, and this is done in order to produce high-quality vaccine. Some of the techniques used in antigen purification are the same ones used in protein purification. For example, stuff like separation that involves differences in size, binding affinity, or even biological activity. The fourth step, addition of other components, is done so we can have our purified antigen survive for as long as possible. The last step, packaging, of course, is self-explanatory. The vaccine is packaged in a vial or a syringe once it's ready. The smallpox vaccine was created by a scientist named Edward Jenner. He injected material from a cowpox virus into an 8-year-old boy. The experiment, fortunately, was a success, and the boy never developed the cowpox disease. This experiment was the basis of smallpox vaccines, and since then, we've managed to completely erase the pathogen from existence, meaning we've managed to completely erase smallpox from existence. As for my final comments, I would just like to mention that vaccines have had a tremendous impact on all aspects of mankind. However, there is still so much work to be done. According to UNICEF, 1 child dies every 20 seconds from a disease that is preventable by vaccines. So of course, we need to do a better job of making vaccines available to all of those who need it. And not only that, we also need to stay focused on developing new vaccines that can prevent us from other diseases like HIV, malaria, and even Ebola.